Genetic engineering. It's a fascinating topic. In this video series, you're going to learn about the ethical considerations and usage of biotechnology when it comes to forensics, medicine, agriculture, and a variety of other applications that we can apply it to, even in medicine. So biotechnology refers to any technology that's used to change the genetic makeup of all living things to make products. These products are things like antibiotics and vaccines. We even see now in synthetic hormones and biofuels and a lot of food production. When we're talking genetic engineering, the direct manipulation of an organism's genome, it's all of the genes contained within its living system, its living chromosome system, using biotechnology. Okay, so we're going to directly manipulate the organism's genome, its normal genetic makeup. And this process usually replaces a specific gene in an organism in order to ensure that, the organism is expressing a desirable trait or a trait that we are, trying to either manufacture. Recombinant DNA, that's artificially made DNA. On certain organisms, we may know where a specific gene is on a chromosome, and we might want to artificially make that DNA and be able to splice it out of one organism's genome. So it just sounds so sci-fi, but how can we even do this? All right, so a genome is an organism's complete set of DNA. That's all the genes. Like we have the human genome, and that's all the genes that make up a human. It was a project back when I was in high school, and it was really kind of first completed in 2003. There was this rudimentary genome map, and we had the entire human genome coded. We knew what chromosome had insulin protein, what chromosome had blue eyes, what chromosome had, webbed feet, or whatever trait that we were looking for. So after we figured out where all the genes were located, the ENCODE project was launched. And it was kind of like a follow-up to this, but it was really used to interpret the sequencing from the actual Human Genome Project. So now that we know where the genes were, how can we interpret the sequencing? How are they pieced together? How are they puzzled together? So an even more detailed genome map was needed. And so we're calling it the 1,000 Genomes Project. That was the even more detailed genome map, which we sequenced over 1,000 different people from a variety of ethnic backgrounds. And that took a better part of a decade, but it was completed here recently in 2015. And what we were trying to do is get a whole variety of genes that were located on different chromosomes. So sometimes different ethnic groups, different races can have the same gene. We are all humans, we're all the same species, but it can be phenotypically expressed differently in different ethnic groups, races, and just different people. So we wanted to know how much variety that gene had within the human genome. We were basically looking for genetic variants within our human population. I want to say it was 25 different communities were sequenced, or populations. And a lot of them were isolated populations, like Bengali and Bangladesh, and isolated Finland population, and then maybe a Peruvian population. Humans created this wonderful gene map. It's how we communicate what genes are on which chromosomes, and our findings that we got from different parts of the world, because it wasn't just the United States involved in this Human Genome Project and this detailed mapping and genetic variant sequencing stuff, but we had a map, and so we would basically lie out each chromosome and be able to tell which type of gene coded for what type of protein and where it was located on chromosome number 8 at location 145. So how do we get those little pieces of DNA out so that we can figure out what they are? Enzymes cut DNA we can use what are called restriction enzymes to cut strands of DNA at specific locations. These, how it knows the site is probably for a later time, but we have restriction sites. There's a combination of DNA nucleotide sequencing that one particular enzyme will look for and cut the DNA strand right there. So when it cuts at a specific site, the resulting fragments have at least one sticky end, and it can be joined with other DNA fragments by DNA ligase, and ligase will build the DNA pieces back together. And we use this type of pattern to create recombinant DNA. And then we can tell like what specific combination of genes are occurring where. PCR, or polymerase chain reaction, is a technique that allows you to copy a piece of DNA without actually having a cell. So we want to create more quantity of a piece of DNA before we can use a restriction enzyme on it, or start to piece it together or run it in a gel. This is also known as like DNA amplification, and we can make lots and lots and lots of copies of that sequence for different types of tests in a very short amount of time. Once we have a lot of copies of that DNA, we can use a restriction enzyme to cut the DNA at a specific location, and then use a laboratory technique called gel electrophoresis. It's a method that we use an electrical current inside of a gel, and it sends the DNA fragments from one side of the gel to the other. And if you can imagine this as larger pieces of DNA, they don't travel quite as fast through the gel. As smaller pieces of DNA, they will travel rather quickly through the gel. It's also called DNA fingerprinting. From this fingerprinting, we can get a pattern or a sequence of known allele combinations or base pair size. We at least know that we can compare pieces of the DNA because they've been cut at the exact same nucleotide sequence site. I mean, there's lots of different types of examples of genetic engineering. I mean, these days, genetic engineering and biotechnology can be applied to use for all kind of things that benefit society. We are going to look at just like the personal genome sequencing, the Human Genome Project, and also cloning, and also how we get bacterial transformation, which is very cool, and then gene therapy. So sometimes in order to be unfortunately diagnosed with a disease or to determine if you have, reproductive vitality, it's important to have your genome sequence. We use that DNA sequencing technology to have your personal genome sequenced. This provides us lots of information about you, like your physiology, like what you're susceptible for disease, certain diseases like, cancer. These days we can have your genome sequenced and run a panel to find out if you have 20 to 20-ish to like 170 different known genetic alleles that are used to cause breast cancer. And so these can be, this can be particularly useful. It's very expensive, but it can be useful. And it also helps couples who can potentially carry a genetic disorder with vitro fertilization, because if you carry a genetic disorder, especially if it's terminal, you don't want to pass it on to your offspring. So if we know where that genetic disorder is located, we could potentially use a sequencing technology to find out where it's at, and then from there use special reproductive techniques to ensure that your new offspring will not have that genetic disorder. I remember the year where they cloned Dolly. So gene cloning is, yes, we have cloned sheep, several animals and stuff actually by now, but cloning also does have lots of other useful things, even in terms of medicine and in terms of agriculture. But, gene cloning produces multiple identical copies of a gene, and then transformation is the process of inserting that recombinant DNA into some host cell. And remember, recombinant DNA has two species or two combinations of DNA inserted in there. So recombinant DNA can be inserted into a bacterial plasmid, I should say a bacterial plasmid is what we call the DNA strand inside of a bacteria. And then when the bacteria just reproduces like normal, it makes millions and millions of this cloned gene itself. So we can take one bacteria cell and insert a piece of recombinant DNA and then get that bacteria cell to reproduce itself, because bacteria grow and they reproduce, and it'll make lots and lots of different copies of that exact same gene just through its normal replication process. It's particularly useful when it comes to making insulin. So insulin is a protein that's made by your pancreas, and it regulates your blood sugar. And people with type 1 diabetes, they don't make enough, or basically they don't make any of that insulin. And so we usually have to inject it into the person to kind of control their blood sugar levels. So this type 1 diabetes is often something that you're born with. So we can use bacteria to make and bacterial transformation process to make artificial insulin. So a piece of circular DNA or a plasmid is extracted from a bacteria, and then we use those restriction enzymes to cut the DNA and take out a piece of it. That gene for human insulin is then inserted into that bacterial plasmid, and then we put that bacterial plasmid back into the bacteria. And this genetically modified plasmid or bacteria DNA strand is introduced into those new bacteria cells. Then they're going to do what bacteria do, which is just divide. They divide rapidly, and they start to replicate that DNA strand that was a piece of human, and the bacteria will start to make human insulin, because they have the genetic coding to construct that protein. When we're talking about gene therapy specifically, that's inserting a normal gene or an edited version of an existing gene to fix an absent or abnormal gene. CRISPR gene editing is a technique that helps us kind of understand the genetics behind like diseases. It doesn't just introduce normal genes, it actually repairs the existing abnormal genes. And I should probably mention that this type of repair is sometimes something that happens naturally when that first part of fertilization occurs. But we can use these editing techniques to get behind the disease and start to actually edit the DNA inside the human. You may have heard of stem cells. They've been in the news a lot lately. These are undifferentiated cells that have a potential to become anything. Most of the time they're found in embryos, but they can be found in adult bone marrow too. And we can kind of genetically manipulate them and induce them or introduce them into like places or tissues that have been defected. Usually this is due to some sort of damage or disease, muscular disease. And those stem cells, when you introduce them into that area, because they're at a point in their cycle where they haven't really decided what type of a cell they're going to be. Once they're introduced to a diseased or damaged area, we can kind of genetically manipulate them to replace that tissue or to become new, different types of tissues.